So first I'm going to go through introduction to the localities, the geologic context, and my research question and approach I'm using. Then I'm going to get into like some relevant background of what you guys need to know about our area, and then I'll get into the specifics of my research project. So introduction. So fossil conifers from the Eocene Patagonia are really interesting to study, not only because they are really sensitive to climate and they are these really important environmental indicators of a rainforest biome, but also because they have these really interesting ongoing histories. So when we see these are fossils on the left that are from genera Dephnecarpus, Agathis, and Poplocetras. Those are genera that are only found living in Australasia region today. And then the ones on the right are two examples of conifers, genera Retrofilum and Ericaria that actually have a distribution that's split today between South America and Australasia. And these fossil conifers come from the Eocene caldera lake beds of the La Luchera Formation in Argentina. And the fossils that I'm going to be talking about today come from these two sites, Rio Pichu and the Luna del Mundo. And Rio Pichu is located just outside Bariloche in the Rio Negro province of Argentina. And then Laguna del Mundo is in the neighboring province of Chubut. And we know that Rio Pichu is 47.7 million years old, and Laguna del Mundo is 52.2 million years old. And these dates come from Argon and data enriching dating finite asphalt tuffs. And those are part of archillated magnetism from the European Bluebelt. And having these really great geochemical dates on these fossil formations is really important because it allows us to place them within the geologic context. And it turns out that these ages are really important because they span and just follow the early Eocene climatic optimum. So here we have the famous Douglas showing the global deep sea oxygen isotope record. And what we know is that during the early Eocene, it was the longest sustained period of warmth in the Cenozoic. And so that was from about 53 to 49 million years ago. And so the Laguna del Mundo site is right within that optimum, and then the Rio Pichu site is actually just following that event and in those initial stages of the cooling trend on the Earth. So not only that, but tectonically it's a really important time in Earth's history because of the connection between South America and Antarctica. So here we have a map that's showing a paleo reconstruction for this time period of 56 million years old, and the sites are located there at the paleo latitude of around 45-ish degrees, and the modern coastlines are shown in black. And during the early Eocene, actually throughout the Cenozoic, until that point, there was this close connection in which we know that there was biotic interchange going through South America and Antarctica. But we know that from lots of different evidence, especially fossils of mammals and plants. And it's thought that by the Middle Eocene, the eastward shift of the Antarctic Peninsula really began, and the Drake Passage began to deepen. And this is just one framework that we're using. It's a really complicated thing to know the exact timing of that event, but this is what we are using. And so, given all these like tectonic changes, climate changes, I'm using fossil conifers as a model system to see if there's evidence for floral turnover in Patagonia during this time period. And so I'm using the approach of systematics in order to address this research question. And if there's one thing that I hope you guys take away from this talk is the importance of looking at biology in order to inform like, the geologic maps. And so systematics is really just studying that biological diversity and evolutionary history. And there are these two different parts of it, taxonomy, where you're actually describing and naming things, and then the phylogeny aspect, where you're putting those organisms in an evolutionary hypothesis to see how they're related to one another. And this approach is really good for answering the question because in order to see if there are changes in the flora, so to find turnover, we have to actually know what things are with certainty to know if they're the same or not. And once we have classified those species, we can then compare their past and present ranges. So getting into the paleobiogeography, so comparing between the sites, between the continents, where they are now, where they were then, and so I'm hoping what you guys are also going to see from my talk is that systematics is not an easy thing. It takes a lot of time to actually go through each individual species. And these two localities have both over 100 species at each. These are angiosperm dominated flora. And so collaborators have begun to work on the systematics of these different species. And there is some preliminary evidence in the non-conifer taxa that there are certain species that are only found at limited limits. So as these researchers have described these species, they've noticed and they've checked that, well, we don't see them at Rio Beach Lagoon. And this is something that hasn't really been well studied yet, and I'm actually going to be studying that for my PhD here. But I just want to point out a few of these key ones. So there's cycad, fern, eucalyptus. A lot of these are ever-wet taxa that really thrive in these environments that have to be wet all the time. And so if you guys want, after I can give you a table that has all this information with the citations and the species cited there for you. But this is just kind of illustrating what's going on. So now we're going to get into the background. So Araucaria are this really iconic genus of subtropical tropical conifers, and they're from the family Araucariaceae. So this is a phylogeny, so it's an evolutionary hypothesis that's showing the relationship among the living conifer families. And when most of you guys think of conifers being in the northern hemisphere, you're going to think of like pine trees, fir trees, spruce trees, and things that are in that pine family. But as you can see, there's actually a really large diversity within the conifers, especially those that actually only occur in the southern hemisphere, as you can see. And so this is basically generated by taking morphological and DNA data, creating a phylogeny, and then the support values that you see show that this is a really well accepted. We have a good understanding of how these groups are related to one another. So if we zoom in on Araucariaceae, you can see that this family consists of three genera. So there's Araucaria, and then there's a sister clade of the agathidores, which consists of agathis and Willemia. And Willemia is a interesting case because it is a single species that's represented by this genus, and it was actually discovered 25 years ago in New South Wales in the remote rainforests of Australia. And Willemia has really interesting foliage that is twisted. It's four-ranked, and the leaves are sub-opposite to opposite. And then we have agathis. Agathis has about 17 species. And as you can see, the leaves are also sub-opposite to opposite, but they're very, they're broad and they are oval to elliptic in shape. And then we have Araucaria. Araucaria has 20 species, and the leaves of Araucaria, in contrast, are helically arranged around the branch, and they're more like slender and needle-like. So you might also be noticing that there are these subdivisions just within the genus Araucaria. So there are these key morphologic and genetic differences that separate Araucaria into these four groups that are called sections. And you can kind of think of them as like subgenera. So the four sections of Araucaria are Intermedia, Bonia, Araucaria, and the taxa. The taxa has 16 out of those 20 species. So as you can see, these other three sections either just have a single species, or in the case of section Archaea, there are two species. And I have up here living material of the leaves. So some key differences to note is that section Tacta is the only one that has univate leaves. So the other three sections, the leaves are multi-vate. In between section Tacta and section Intermedia, these are the only two that have what we call dimorphic leaves. So there's a difference in the morphology of the leaves depending on if they're juvenile or mature leaves. And that's shown here for the Tacta, the two different ones. And then we have examples of the cone scales. And the cone scales are what make up the seed cone, the individual components. And one thing that should jump out right away is that section Archaea lacks any of these like things on the side of the seed in the lateral extension compared to the other three sections. Section Bunya, it's got this really woody, lignified cone scale, and it's actually the only one where the seed becomes detached. So in the other section, the seed actually stays within the tissues of the cone scale. And then between intermediate and tactile, you can just see that these ones are like way bigger and it's very large and thin compared to section tactile. So Archaea are entirely restricted to the southern hemisphere. So this is showing the modern biogeography of those sections. And as you can see, section Archaea is the only section that's outside of Australasia, and the rest are all in Australasia. So section Bunya is a single species in Australia, section Intermedia, a single species in Papua New Guinea. Section Tacta, actually 14 of the 16 species are all on the island of New Caledonia. And then you have just one species that's on Norfolk Island, and then another species that's found in both Australia and New Guinea. So we actually went to the forest in the Andes in March of 2017, where we got to see some of the section Archaea growing there. And so hopefully you're starting to recognize the trees on the landscape. They have that really cool candelabra-like shape. And they have these really crazy leaves that are very sharp and spiny actually. And that's where that common name of the monkey puzzle tree comes in, because they would be really hard for a monkey to try to climb. And what you can see over here are those seeds and how they would be arranged around the central axis into forming that cone before they were dispersed. What's cool about these seeds is that they're actually really important to the Mapuche Guen people of the Andes, and they actually rely on these for their diet. And it's a really interesting thing. We actually tried roasting them and eating them, and they're kind of good. They kind of taste really like starchy. I guess you can think of it as like a giant pine nut, but not. And then it's also really cool to me because not only do the people in the Andes think that these trees are sacred, but also in Australasian cultures, they also really value these trees, so it's really cool. So I did not get to go to Australasia, but I did go to the Montgomery Botanical Center to see the cultivated versions of section Chunka. So here you can see there's like tons of cones that are growing on this tree, and I cut them down so I can collect them, and you can see how different the cones are. It's really resonant, so you can see like the white resin coming out. The leaves that are very small compared to the section Archaea, and these are the male cones that have the pollen. So in contrast to this really reduced distribution that they have now, Archaea were once found worldwide during the Mesozoic. So we have an amazing fossil record of these. And so these are some of the best examples from the Mesozoic. What you're going to notice right away is that I have a lot of these anatomically preserved seed cones. So these are really cool because they have a lot of the features that we can observe that really help to know that they are members of Archaea, and so they're going to come up later. What I have here in this corner are two that are within Archaeaceae, but they're actually agathioid fossils. So they're more related to Agathis or Wallonia than Archaea. And those are really important ones that are going to come up later as well. And I threw up a picture of some leaves as well because there's tons of other fossils besides these that can be just like a single cone scale or lots of different foliage. So we have a really rich fossil record. And after the amputation, there's basically a preferential survival of this group in the southern hemisphere. So they basically went extinct, as far as we know, after the amputation in the northern hemisphere. And what's really interesting is that we have this complete fossil record, and yet the molecular clock age estimates for the age of the lineage is actually very young. So molecular age estimates are basically taking a phylogeny and then they use different algorithms to try to calibrate that tree that results and try to put dates on the different nodes to understand when lineages would probably have evolved. So the crown is basically saying the most recent common ancestor of that group and all the living and extinct descendants. So here you can see some of these estimates. So for the crown of the family, they're as young as 36 million years old, 65 million years old, and that really contrasts with all the things of fossils. And so one key thing that we have in the fossil record are those two agathoid seed cones. And what they tell us is that because those two were there, that means that the split, the family, okay, so because the agathoid clade and the Araucaria clade has split by that time, the family must be at least as old as those fossils. So that's where we get this idea for the minimum age based on fossils. So then, of course, having that young age of the family itself affects all the nodes within the family. So for section Tacta, for example, this is thought to be a really young group, and we just don't really know for sure if we have a fossil that represents the section Tacta crown. So remember I mentioned that there could be just isolated cone scales, isolated foliage. It's really rare to have all the evidence you need to know if you have a living section. Because remember when we went through the living section, you have to look at the leaf characters, you have to look at the seed cones, and so it's not as confident when we have this. And the problem that comes in with these molecular age estimates is that they basically take like 5 fossils to calibrate the tree, maybe only 2 of them are from Araucaria, and in the example of this date from Venezuela, they actually use a Jurassic cone scale that they think is from the Chaco, but then they still get estimates for the crown that are really young. And so what happens is it's like where they place these fossils to calibrate the nodes is often in question, and then also algorithms they use, so it's really complicated to understand why this happens. So now I'm going to get into my study. So if you think back to the limit side, or Yokuchifu, here I am in Yokuchifu, collecting all these awesome fossils from the lake bed. And my research is really focusing around this fossil, Archaea pichileuthensis. So this was a fossil that was described in 1938 by Berry. He published these 6 fossils in his description, and he really noticed that these look like that Australasian section Eutacta, but they have not been really studied in depth since 1940. And so he thought that these looked like section Eutacta because it had those slender leaves with those little needly leaves on it, or slender branches with needly leaves. He had these broad cone scales with the wings on either side of the seed. And this is a really significant result if it is true because, of course, this section only lives in Australasia. It's not anything like what lives in South America today. And it's actually really highly debated about the origins of this lineage. Did this lineage end up in Australasia as a result of tectonic carriers, or was it more dominant pattern of long distance dispersal across oceans? So this fossil could potentially speak to that for this lineage. So in addition to that significance, there's also those molecular age estimates. So if this fossil is actually in section Eutacta, then it would be much older than those post-unwanted molecular age estimates. So this fossil has the potential to really test those estimates and maybe inform them in the future as a calibration. And so what we have now is this, we've gone and we've collected so many more fossils. There's a huge fossil data set that now has pollen cones for the first time that have never been described. We have fossils that are like very exceptionally preserved, and it's really rare, again, to have all of these ornaments present. And we have leaf cuticle and pollen now that we can actually use microscopes that they didn't have back then in this level. And these fossils are coming from both the type locality of Great Acacia Food and from Laguna. So we have really large collections, and with these collections we can test the following hypotheses. Um, the first one is that the nearest living relative from these two Patagonian sites is in fact the Australasian section Tacta. And the second hypothesis is that there could actually be a difference in the species between the two localities. So my data consists of a bunch of herbarium specimens, a really large fossil data set. You can see the breakdown here between the sites and between organs. And then there's this combined matrix. This matrix was originally published by Scott et al. 2015 in a study of Archaeocariaceae, and then it was subsequently modified last year by Scott et al. 2018 in a study on Agathis. And so what this matrix is it has a morphological component and a molecular component. So the morphological part has 62 characters. 10 of the characters are continuous characters. So think of like leaf length, leaf width, things that have a minimum and a maximum measurements that were observed. The rest of the characters are discrete characters. So ones that can be coded with 0, 1, 2, things like that. So for example, attachment to the pollen cone, terminal versus axillary. And then these characters are coded for 32 living species of an in-group, as well as 12 fossil taxa. So the fossil taxa, I'm adding my 2 fossils. The other 10 fossils are 6 of those Mesozoic Araucaria sea cones that I showed earlier. 6 of those. There's those 2 agathioid sea cones, and then there are 2 agathis fossils. So one of them is an Eocene agathis actually from these 2 same localities, and then the other one is the oldest known agathis macrofossil from the Paleocene, also from Patagonia. And so I added my 2 fossils, and then I did not change anything with the molecular data, which consists of basically 23 genomic regions, mostly plastid, but also some nuclear mitochondrial DNA. And those are included for the living species, and then a really large outgroup of 306 species. And that outgroup basically serves to polarize all the characters and to really help us get as best resolution as we can within Archaeocariaceae. Okay, so in my method, so first I have to go through all the literature, so basically all the publications that describe the living species of Araucaria. And then I went through the herbaria and living collections to really understand what defines this living section and the diversity within it. So in addition to going to the Montgomery Botanical Center, I also went to the Huntington Gardens and Library, and I studied their living collections of several cactus species and their really large herbarium research collection of species, of these types of species. And then here I am at the Harvard Herbaria, studying their really big collections, taking a lot of pictures of all the specimens for my research. And when I went to Montgomery Botanical Center, I also made collections there. The different species I had that were coning, so I have reproductive material for them. And I made these voucher specimens that I deposited at the Penn State Herbarium so that I can reference them in my thesis and kind of like leave a legacy here for other people to see exactly later. So from all this work on the collections, I got this idea of all the different features that I was going to look for in the fossils. So these are the key things about Ithaka. We have the dimorphism in the leaves. So younger leaves, they're more linear. They get more incurved as they get older, and they actually get wider in width as they get older. And then you have these pollen cones that are attached terminally. So at the end of the branches is the only section that they do that. The rest are auxiliary, so like coming off the side of the branch. So the pollen cones, they're these like cylindrical cones. They have these, what you can kind of see of those like diamond shapes. They're beautifully arranged, pointed microsporicular heads. They have these basal bracts that are very long, slender, triangular leaf, modified leaves that start at the right of the base and then come along the cone line. We can also know from leaf cuticle, section Mutata usually has stomata that are oriented perpendicular or oblique. And leaf cuticle is basically the skin on the leaf where the stomata are like those gas exchange pores. And between different species, you can have different cuticle patterns. And then, of course, we have those seed cones with the hundreds of individual cone scales that are spirally arranged, and those cone scales have those really thin, papery wings on either side of the tube. So once I have a good idea of what Intacta is, I can go and study the fossils. So I had to go to Argentina to prepare them in the lab, so taking away any extraneous matrix, imaging them with high resolution macro photography, and then studying them under the microscope and checking them under epifluorescence and taking more photos under the microscope. And once I have this giant photo library, I can take it back to Penn State and I take all the photos in Bridge and Adobe and I keyword them by these different features to organize them, and then I measure them mostly in Photoshop. So then once I have all that information, I can actually work on the descriptions. And once I also have that information, I can use it to code them into the matrix. So again, I'm using that Stockyl 2018 matrix, and using a combined approach, combining the morphology, combining the DNA, using maximum parsimony method to create my tree and test my hypothesis. So here you can see working in Mesquite, which is the software I use to code and organize the matrix. Here in TNT, that's where I actually ran an analysis and get my results. So now we're going to get into the results. Okay, so then my key results are that I was able to confirm the relationship of Intacta to these fossils using both the taxonomy part and the phylogenetics. The second thing is I was able to distinguish two different species from these two different localities, detecting the turnover between the sites. So if you look at my measurements just quantitatively at the basic length and width, we can see that the measurements of the fossils overlap with the living section Intacta. So in these pictures, I just put an example of where those measurements come from with length and width. And then you can see that the fossil taxa are in the colors, and then the extant taxa, the measurements come from these two taxonomic sources. And I want to point your attention to the range. And what's interesting is that they are always at the smallest end for both organs, and that's something that's been noted in the literature, both for this guy in particular and for fossils of Uruguay in general. Fossils tend to be smaller than the living material. And then for these pollen counts, for each site, I just had one complete one that it had a full length measurement. So that's why they're represented as stars, and it kind of limits the ability to compare them because you can see like the big ranges of sizes that they can be for different species. So now I'm going to get into the actual taxonomy. And at the top left, you have that example of the modern material again. And so the first thing with these leaves is that they have the helical arrangement of those leaves, so we know that they are Araucaria. Between the two, they also have dimorphism, so we see these like kind of two different end members in the fossils. And in Rio Beach Lagoon, that first type is more like cellular and spreading. You can see that they're like spreading leaves, and then in Leguminonco, it's more like acicular and falcate. So these are more needly, they're more curving. And then in that second type, in Rio Picture food, they're more lanceolate leaves, and they're more address. And then with the Leguminonco leaves, those ones are more ovate and imbricate. So there are these subtle differences between the leaf morphologies that I've noticed, and it's something actually with the living taxa species and different species. So when we look at these under the microscope, though, we get a really good piece of evidence to why they are two different species, and that is because they have different orientation of the stomata in the fossil cuticle. So what you can see is in the Bononco, they're oriented perpendicularly in these rows, and then in Ramphasia 2, they're oriented parallel. And so that's a really big difference between the two localities. When we look at the cone scales, again, we have that characteristic single embedded seed with like the two wings on either side that leads us into tackle. But between the two sites, Rio Peach Lafu is actually a bit larger in size, and they're also have these just like wider wings. They're more triangular at the apex, I find, and the Linodongo, they're more rounded at the apex and overall smaller in size. So in order to try to quantify those differences, I have these box plots. So the Ericaria mensis is in the gray, and then the species from the Linodongo is in the white. And so you have like the median is the gold line, the median is the plus, and then these are the quartile ranges. And then you can also see like the t-test results over here. So they were all statistically significant. And you can see that difference here between the total length of the two and the total width, with each one being bigger. And then when you put those into a ratio, you can also see that the signals on those tone scales are closer to a ratio of 1. And when you look at where that position of the max width is, so on the scale, at how high the scale is that maximum width occurring, it's generally around like half of the total length versus higher up on the luminal. So then when we get into that really critical new piece of information of the pollen cones, we have this amazing fossil that has this terminal attachment of the cone to the branch. And this is a really big deal because it allows us to know that these were part of the same plant. So like all those isolated branches, all the isolated cones, like we're from the same thing. And another important thing is that fact that Utaka is the only section that would have those terminally attached cones. And actually, when those cones are mature, they usually just fall off the tree and become dispersed. So we have lots of, well, not lots, we have a few examples that have those dispersed isolated cones, but they have those basal bracts that are really important to know that they are Ericaria. And we also have pollen that I found like, in this cone that is characteristic of Ericaria. It's just kind of nondescript, but it's an inarticulate pollen. So then when we get into the test or the phylogenetic results, what we have here is a look at the Ericaceae family. The fossils are the ones that have like the asterisk after it, and you can see my fossils in this study are in red. The agathioid here, you can see the two agathioid fossils are coming out in the stem position. Those are those two seed cones that I was talking about, and then there's the lamia and agapus coming out in the crown. And so I just marked S and C for stem and crown. And these results for this plague are the same as what was found by the previous study. I didn't do anything to change that, so I'm just showing it kind of for completeness. And then you have Ericaria. So you have all of those 6 Mesozoic seed cones coming out in the stem of the genus. And then you have at the crown is where those four living sections come out. And at the stem of the taxa, that's where the Rio fossil comes out, Archaeopterygium. Then you have the crown, where these two fossil, or these are two living species. Penicillium is that one that's found in Australia and in New Guinea. Heterophila is the one that's from Norfolk Island. And then you get the New Caledonian plate of all the New Caledonian species. And interestingly enough, the Laguna species comes out within the crown, within the New Caledonian plate. And so, in terms of differences to why they're not both in the crown, when you look at the matrix, how it coded them, really the only difference in the scores is the somatal orientation. So if you remember that difference where the Rio Pichufu actually had parallel orientation stomata, that's a character that's only seen in these other sections. So that can be pulling it down over there. And then other than that, Archaeopterygium is the one that had that terminal attachment and had that pollen stored in. Those are just left blank for the Laguna species. And then, of course, the continuous characters between them vary, although they were not super different. They did have overlap between the regions. So those are the types of things that I'm thinking about to see why they could be different. So getting into further discussion, so this presence of section taxa adds to this knowledge of the other species that have been described for the fact that New Guinea is this really great modern analog for these eastern Patagonian forests, especially within the Gillico. Because you have living Araucaria section taxa, Agatha, Staphylocarpus, Papicetris, Papamausis, Eucalyptus, Denisoma, like all these genera that we find there are literally living in the Guinea right now. So it's really cool that area section drought resistant, adapted species, and they can actually grow in these dry rainforests that get less water than regular rainforests. And so what we're thinking here is that irrigated defenses may actually be better adapted to the changing soil environment. So if you remember, the forest is going basically from this very ever wet, aseasonal environment to this cooler, drier, maybe more seasonal environment in the future food. And so what I've been trying to check into literature is looking for anything that can tell me about what kind of adaptive traits Araucaria defenses might have that could show evidence of that. And I did find at least one study that suggested that the somatal patterning could be linked to climate change, and that's because, especially seen in drought-tolerant species, that somatal patterning actually originates from the relationship of the surrounding cells and the guard cell, the physiology between the two. So this is something that has not really been studied in depth with conifers, but I think that there could be a connection there. And then what we know is that eventually Araucaria went extinct in South America. There's actually only two other fossils that come after the Neoplacian at the time. There's one from the early Oligocene of Chile and then one from the Miocene of Chile. And then after that, we don't see any more. So in conclusion, what we have here might be the most complete evidence of section taxa in the fossil record. So there's only a very select handful of maybe 3 examples in the fossil record where you have all 3 organs preserved. So this is really strong evidence for the section. Then we have the fact that these fossils are about 30 million years older than those molecular estimates of the lineage. And not only that, but they're from Patagonia. So it kind of puts forward the idea that these were Gondwanan lineage initially, and maybe people need to consider more as kind of experience of the taxa thing, especially because of the position of the Laguna species within that New Caledonian plate. And not saying that both didn't have an effect on this but just don't discount the claim. And then it also is showing that added evidence to what we know about the change of the rainforest between after the early 1879 optimum as December 2. So with that, I want to thank Nacho and all the people from collaborators in Patagonia that made all this work and possible. And I want to thank all my funding sources. And thank you again for coming and taking questions.
Thank you, Gabby. Would you like to play on or?
That's fun.
So at this time, the public may ask questions of the candidate.
So you, one of your photographs, you had a fossil of one of the plants, if you remember which one, but it was in England, like in England, and you had said that they, have been extinct from North American continent. So could you find these presumably in the United States, these fossils, or are there hotspots for them in the northern hemisphere?
There are a few for sure throughout the Northern Hemisphere. So there's actually a Jurassic cone scale from Massachusetts that's been used in the molecular calibration. And so we do have Araucaria in the United States.
In your character matrix, did